Right, Gracie, what I'm going to do is, obviously with this video, I'm just going to go through some of the stuff with 10.2, but you may need to go back to 3.4 to look at the inheritance there, because this is building on from that. So if you've got your textbook, maybe read through that. A lot of that is very similar from GCSE, so you remember your homozygous and your heterozygous and your phenotype and your genotype. If those words don't ring any bells, then have a look back at 3.4 and then watch back at this video. So what we're looking at today is we're looking at dihybrid inheritance. So it's all to do with where the genes are located on the chromosome. So generally, unlinked chromosomes are, or unlinked alleles, they're not going to affect each other. So what we'll do is we'll try and look at an example of that in terms of Mendel's P's. You may want to, if you can't remember about that as well, there's some resources on the classroom for that. I'll try and show you those in a little bit as well. So you should remember from meiosis that you get random orientation. So that's how these are going to like randomly orient themselves when you're going through division. So obviously you've got your homologous chromosomes here. Now we've got them. So you've got your sister chromatids, but it depends which way around you've got them. So you can see this top one in this time, they're over this side. So it's going to lead to a different combination being formed in the gametes. And then obviously when fertilization occurs, then you're going to get pairing up as well. So you've got random orientations to how these orient themselves during metaphase 1, or how once they have then sorted, then you've got the independent assortment. And that refers to the behavior of alleles of unlinked genes. So we'll give an example of what I mean by unlinked genes. So if we've got them here, so you see these are on different chromosomes. So A and B, they're not on the same. If they were linked, they'd be on the same chromosome, but they're not. So we can see here we get different combinations of these different characteristics. So this could be like for eye color, this could be hair color. They'll be on different chromosomes. So we can get A going with that big B, or we can get A from here going with this big B, or this A with that big B, or this A with that. Okay, sorry for the interruption there. Just with the other class as well. So we've got the idea of here of how the meiosis is going to lead to that random or that independent assortment. So, and as Mendel says, the presence of an allele of one of the genes in a gamete has no influence over which allele of another gene is present. So A is not going to affect the presence of B. A can only affect the other A's. Okay. So how does that work in practice? Well, what we've got here is what we call dihybrid crosses. So when you did this in year 10, you think about it, we just did monohybrid. Now this looks more complicated than it is, but it's really quite simple. So mono is just one. So you remember if it was X, Y, X, X, you could do those crosses and it would only use these. Here we're going to have an outcome of 16 possible outcomes. So if this is heterozygous at both loci and this one's heterozygous, that means we're going to have these combinations, aren't we? And we know which one's dominant and which one's recessive, because dominant is going to be the bigger letter. So if we've got two smooth yellow peas, they would have the phenotype, they would look smooth and yellow, but their genotype would look like this. So you can see you've got the big S, big Y, small y, big S, small s, and so on. Now if we were to cross these, then what would happen, we're talking about the different combinations. So the first combination we've got is we can have a big S and a small y. So that so I'll go into that first box. The next combination we could have is a big S and a small y. Then we can have a small s and a big Y, and a small s and a small y. So they're all the different combinations that could possibly be in the gametes of this P. Because this one's also heterozygous, we'd have the same combinations here, and then we bring them together. We can then look at the genotypes that we could possibly get from crossing these together. And if we've got a big S, then we know it's going to be smooth. If we have a big Y, we know it's going to be yellow. So obviously anything that's got a big S and a big Y is smooth and yellow. So we can see all these top ones are smooth and yellow. But then here, this one's going to be smooth and green. This one's still smooth and yellow. This one, smooth and green. Smooth and yellow. Then here, rough and yellow. Here, rough and yellow. Smooth and yellow. Smooth and green. And then we've got rough and yellow, and then finally got rough and green. I think I've said that right. I may have said this one's smooth. Yeah, smooth and green here. That's smooth and green. That one's smooth and green, and this one's smooth and green. So if we look at the actual phenotype, the ratio of those, when we look at the ratio coming out at the other end, you can see here we've got 9 smooth, 3 smooth and green, and 3 rough and yellow, and 1 rough and green. Now, if we did this with others, we could do various crosses. I'm just trying to find the example of where, if we didn't know what it was, we would cross it with a homozygous recessive if we were trying to work it out. So if we didn't know, a rough pea, yellow pea is tested to determine its genotype. If we're doing a cross, we always cross with a recessive to find out its genotype. So if we don't know, the rough and yellow could have these different phenotypes. So we would always do a cross test with a homozygous recessive. Homozygous recessive is always going to has to have the 2 lowers. So we don't have to do the whole lot because we know it can only have that genotype. So it has to be that. So then we've got, obviously if we're trying to work out this rough and yellow could be this, or it could be this. So we can cross all those, cross, and then work out which one, when we look at the offspring, what do we get? So if we crossed it with this, we would get some green peas would be present. So that means the unknown parent would have to have this genotype. But if we didn't get any green peas, then we know it's going to be the opposite genotype. Okay, so what I want you to do now, Gracie, is to go away and look at the book and try and do some of those crosses. If you find it difficult, feel free to email me and just we can talk about that. But also have a read through what's in your textbook and what else is on the on this slide. Okay, it looks more complicated than it is. You could read through about Sooty the guinea pig as well, find out about him, that story there, and how we can look at different characteristics and work out his genotype from his offspring. Okay, so yeah, email me if it doesn't make any sense. It seems more complicated than it is. I don't think it's that complicated, but then I'm a biology teacher, so I shouldn't find it that complicated. If I did find it complicated, that's when you want to worry. But yeah, the only way you're going to do this is by practice. So within the book, on Classroom, on the notes, you should have some information on there that's going to help you. Okay, so just go into Classroom. Obviously the PowerPoint I used was on here, and if you can look at the notes section as well, have a go at those dihybrid inheritance, and then you look down. Don't worry about the chi-squared, we'll come back to that. You may have done that, I need to check on that one. But what I would say is go back down here. Hopefully you've done the formative. So yeah, you sent that to me last week actually. So you've done the formative assessment. I would then recommend you go to inheritance. There are some videos in there about Mendel. So if you open this one up, obviously not there. You got Gregor Mendel. Watch that video, but also on the section here, it's just coming off the screen, but on 3.4 on the notes on the first bit, there is a little thing that goes through Mendel's inheritance, so it's good to have a look at that as well. Like I say, if you've got any questions, then feel free to email me and I'll try and help you as best I can. Hope you're keeping well, and hopefully see you soon. Cheerio, goodbye.